Okay, good morning, everyone. My name is Diane Ward, and I am the Enrichment Chair for the Coordinating Centers of Excellence in Hematology. And this morning, we want to welcome you to our first in a series of monthly Microscope Monday lectures, where we have several experts in the field who run core facilities at different centers of excellence in hematology around the country, who have offered to share their expertise in microscopy to teach us, good microscopy experimentation and the tools that are available and how to interpret your data or software that might be available to interpret data. For a moment, I'm going to just introduce you, if you haven't looked at our website for the Cooperative Centers of Excellence in Hematology. What I'm going to ask you at this point is to please mute yourselves. We'll let Dr. Bewersdorf be the speaker and everybody else mute, and we'll run better. I'm going to just share my screen right now. Let me get it. Okay, so this is our seminar series that you have joined today, and we have one every second Monday of month. We have one break here that may be filled, but it also may just remain a break for us. And we hope you will join us. The Zoom meetings have the same ID number throughout and the same passcode throughout, which is this image CCEH. And we hope that you will share that with your colleagues and let them know about this really great opportunity to learn more about microscopy and the tools that are available at the centers of excellence, but also learn about what microscopy might be the best approach for you. The second thing I want to point you to is our website, which is right here. It's just cceh.io, and it's the Cooperative Centers of Excellence in Hematology. And here you can see our monthly microscope seminar series. We also have a Meet the Experts seminar series. What I want to draw your attention to are two things. This link here, which is the services link, which I'm clicking on, which will tell you about maybe some questions that come into your mind about how I can perturb cells, how I can manipulate cells. And so if you click on one of these links, it actually will tell you about different centers, cores that they have available for you to use for studying hematologic questions. And so each of those cores have different ones. So for example, here's another one that shows you about animal modeling, where you can click on this link and you can see that the Cincinnati Center and Yale and Indiana and the Fred Hutch have all of these sort of animal modeling systems and experimental models cores that you can utilize for your research. And if you have more questions about that, you can go to these websites and learn more. But also, if you click on these links, for example, this is Yale's, you can actually find the individual that you might want to contact to utilize one of these cores. I'm going to go back for a second. Let me just... And also on this website, there's a funding link, which shows you opportunities for obtaining PNF monies. If you scroll down the page, this is a particular individual who received PNF funding, but we have a call out right now for both Type A and Type B grants. The Type A grants are core credits to use these core facilities, where you can apply for funds to, for example, use an imaging core at a particular imaging facility. The Type B, the Type A's are available year-round. You can apply for them anytime. And then the Type B awards, we have a new call open with a due date of August 1st. And these are $75,000 PNF grants for 1 year. And the details of it you can read on our website here. So I'm going to now disappear that part and come to introducing Dr. Bewersdorf, who comes to us from Yale's Center of Excellence in Hematology. And Dr. Bewersdorf is a professor of cell biology, biomedical engineering, and physics at Yale University. He received his master's degree and his doctoral degree in physics training with Dr. Stefan Hell at the Max Planck Institute for Biophysical Chemistry I'm going to probably Göttingen. How do you say that? I'm making an attempt. Göttingen, Germany.
Göttingen.
Okay. And he then went on to Jackson Laboratories in Bar Harbor, Maine. And in 2009, relocated his laboratory to Yale University. He's an optical physicist, biophysicist by training, and he's been a long-time contributor to the field of super resolution light microscopy development and the techniques. And we're really happy to have Joerg with us today to actually share his expertise in microscopy, and we hope that this will be a great opportunity. Two things for you is I'm going to ask everybody to mute with the exception of the speaker. This is recorded, so you'll have opportunities to go back and visit it if you need to. I would ask you to populate questions in the chat. If there's a burning question, I'll invite you to ask the question, and otherwise we'll save them to the end. And a nice part of this series is, besides this lectureship, the speakers have agreed to stay on afterwards to answer any direct questions any investigators have about their microscopy and what they might get advised to use as core facilities. So with that, I'm going to turn the time over to Dr. Bewersdorf, and thank you so much for agreeing to kick this off for us.
Oh, I'm more than happy, and thanks for the introduction and organizing all this, Diane.
Absolutely.
Let me start my presentation. And I need to display settings, swap, and change to the laser pointer. Okay, so this should work. You see my screen? Okay, so let me just re-emphasize a couple of things that Diane just mentioned. So we're kicking off this this seminar series on microscopy techniques with today's talk, and today's talk is really about fundamentals and foundations. You will not see a single advanced microscope image in here, certainly no super resolution. This is really to build some foundation in case you always were wondering why a microscope works a certain way or why you should pick a certain objective. We'll have all these more advanced talks then over the next year, and some will cover just standard techniques like confocal microscopy, but we also cover very advanced and specialized techniques, and you're welcome, of course, to join any of these. And what Diana also mentioned is this 1 hour after the talk, we are really happy to answer any question you might have. So this is not, well, of course, you can ask questions about this particular lecture, but you can also ask about any other thing, whether it's covered by the seminar series or not. Like if you wonder what microscope you should use for a certain experiment, what fluorescent probes we would recommend, any of this, please come. And you can also just call in for that part of it. You don't have to watch the seminar if you already know the particular technique. This is really intended as kind of open office hours if any one of you is looking for some advice on an imaging experiment. So with this, I'll start my presentation, and I apologize ahead of time. I might go relatively quickly through here my slides with the idea that it is recorded, and if you really want to look at something a little bit more slowly or carefully again later, you can go back to the recording. Plus, we have an hour after the seminar where we can discuss any of these details as well. But I want to make sure I really cover quite a few topics here in the next 45 minutes or so. So today's lecture is Optics Foundations of Taking High Quality Images with Your Microscope. And the overview of what I would like to cover is here. First, I'll very briefly just remind you of a few properties of light, like the wavelength. And then I introduce you to the diffraction limit, which limits the optical resolution of a microscope. And in that context, introduce you to the term of the point spread function, which is one which you will no doubt hear if you're around long enough around some microscopy geeks. And we'll revisit this probably in the coming lectures as well. Another limit to the resolution is the Nyquist limit. We also then talked briefly about noise, which is also limiting what kind of level of details you can see in a sample. I'll then switch over to refractive index, in particular to introduce the problem of refractive index mismatch and how that influences the resolution of your objectives. And all that is then coming to a practical advice here. How do you pick the right objective for your application? In the last about quarter of the presentation, I'll talk about color-dependent effects and how you correct for them, and also some image processing problems and the problem of how you separate two different fluorescent channels from each other if they overlap in spectra. So with that, I'll dive right into it. And first, I want to here just show this very briefly here, this characteristics of light. And one confusing aspect that we have here in optics is that light can be perceived as a wave or as a particle. We know it classically as a wave from our daily experience. If you see the rainbow in the sky, if you see kind of any macroscopic effects, this is where we see 3D light as a wave. We know it consists of different wavelengths that we can spread out with a rainbow or with a prism. And the wavelength that we are talking about here is somewhere about a quarter of a micrometer, half of a micrometer, starting around 400 nanometers to about 700 nanometers. That's what we would consider the visible part of the light spectrum. That's what we are using in our microscopy techniques most of the time. But light can also be a particle. We call it a photon in that case. And that is important to know if you want to understand the concept of fluorescence, where a single photon is absorbed by a fluorescent molecule, and then that fluorescent molecule will emit another photon, another light particle at a different energy and therefore at a different wavelength. We'll touch upon that very, very briefly at the end, but this is today's lecture is really about primarily the wave properties of light, and not so much here on the particle side. So talking about the waves now, a couple of properties here I just want to here bring up. The wavelength of that electromagnetic wave that light is about half of a micron or 500 nanometers roughly. It travels at an incredible speed, the speed of light, which is 3 times 10 to the power of 8 meters per second. And it oscillates even faster in a way. It oscillates 10 to the power of 14 to 10 to the power of 15 times per second. So for all practical needs, we as microscopists or we as users here of any microscopes really cannot perceive that speed of light or that oscillation of that light wave. And actually, there's no detector in the world which can really resolve that oscillation of that light wave. So what we in practice observe is the average of that oscillation, or more concretely, the average of the square of that electromagnetic amplitude here. And that we call the intensity of light. And that's what our detectors, like our cameras, our photomultipliers in the confocal microscope, or your eyes, when you look through the eyepieces, that's what you actually detect. So the intensity is typically measured in milliwatts per square millimeter. And you might wonder, how does this relate to that particle of light, that photon? And if you have a typical laser pointer, this will have below 1 milliwatt of power output. So it will have roughly this intensity here. In that laser pointer, you will emit 4 times 10 to the power of 19 photons per second. So individual photon carries a tiny amount of energy. And when you're talking about lamp illumination or laser illumination, we talk about billions and billions of photons. And that's why in practice, we don't observe them, but we just see this average of more or less uniform intensities that are varied here, and which justifies, again, why we can consider light as a wave and don't always have to think of it as individual particles. So light is a wave, and waves unfortunately experience something called the diffraction limit, and this has been observed already 150 years ago by Ernst Abbe, as well as other colleagues around that time, who discovered that you cannot focus a wave to a smaller spot than about half a wavelength. And that is limiting the resolution that your microscope provides. So Ernst Abbe phrased it roughly along the lines of structures smaller than half a wavelength cannot be resolved. I'll explain that in the next few slides, but just want to introduce this general notion here and then show you this equation. You can calculate this resolution, or the fact that you can only focus any light to about half the wavelength here by, for example, calculating this area disk radius, which is the radius of this pattern here going to the first dark ring. And that's about 0.61 times the wavelength. So about half the wavelength divided by this term down here, which is the numerical aperture. And this numerical aperture is the product of the sine of this angle alpha here, which we call the opening angle, and essentially tells us how strongly we focus the light. And the refractive index is the refractive index of the medium through which we focus, and I'll explain refractive index a little bit later. It's worthwhile though, and this term will appear several times throughout my presentation, to know that this term down here is called the numerical aperture, and I'll call it NA throughout the rest of my talk. So this resolution is essentially proportional to the wavelength divided by this numerical aperture. So I already mentioned briefly this Airy pattern. What is an Airy pattern? The Airy pattern is the pattern of intensity that you will observe on your camera or your eye or your retina if you observe a single point object. Think of it as a single fluorescent protein, or a small, very tiny fluorescent bead or fluorescent object emitting light. It will appear blurry on your camera because of the diffraction limit, and that mathematical function that you'll observe will look like this. And we call that an Airy pattern if you have a round lens here. In more general terms, the Airy pattern is the special case of a point-spread function. And that's the more important term here to recognize or remember. So the point-spread function is the intensity pattern that you observe when you image a point. And in any optical system, like a microscope, where we put an object at the focus of our objective, we will observe the point-spread function in our detector on our camera, for example. So this point-spread function might seem like a theoretical concept now, because you say, I'm not interested in imaging single points, I'm interested in imaging a cell. But the point-spread function really describes, if you know the point-spread function, you know how your microscope will behave. And the reason for that is that you can in most cases describe your object as an assembly of independent points. This is intuitively clear if you think of a fluorescently labeled sample. Let's say it's labeled with green fluorescent proteins. Each green fluorescent protein is a point which will emit light and will be imaged onto your camera. Now you have your cell labeled with a million GFPs, or green fluorescent proteins. Each of these will produce one of these points of function as your detector, and all these points of functions now will add up, forming the image of your cell. So if you know how the image of a single GFP looks like, and you know your distribution of GFP in your sample, then you can predict how your image will look like. So at each position of the protein in your sample, you would just put a pattern of your points of function down in your final image. So that also means, if you go backwards now, that structures smaller than the points of function cannot be resolved, because the smallest pattern that you'll see in your image will be that of a points of function. And to bring this point across even further, let's look at the simplest possible object we can think of here, which is just consisting of 2 points. So you can have these 2 points at a larger distance, at a medium distance, or very short distance. And if you now plot a points of function at each of these points, you'll see how these points of functions start blurring until the 2 objects become unrecognizable and inseparable from each other. So if you draw here, for example, the profile in this medium case here, you see that here the 2 points of functions we get here plotted as a blue and a green curve add up in a way where you can barely discern here this gap in the middle. And this is what we would call the resolution limit here, where the minimum of one of these area disks falls into the maximum of the other area disk. And this is again, this equation that I showed you on a few slides back. So this is now explaining the resolution that you get in the focal plane. So in the image plane, the XY plane, if you like, the plane which is perpendicular to your parallel to your cover slip. But you can also look at the axial resolution that you can get in a microscope. And I don't have time to really explain that in much depth, but I want to show you these 2 formulas here. You've seen the top one here. That's the resolution you can get in the XY direction, the focal plane direction. And this is the equation you can get for the axial direction, the depth resolution of your microscope. And there's 2 differences. One is a not so important one, that factor here in front is different, right? This is about 1/2, this is about 2. But the more important one is that this numerical aperture goes in linearly in this equation, but we have the square of the numerical aperture in this equation. And that means that your resolution changes very dramatically in the axial direction, the depth direction, if you reduce the numerical aperture of your microscope objective. So here I show you typical numerical apertures of objectives that you'll find on the microscopes you're using. And I calculated the theoretical resolution that I get here in the XY direction and in the Z direction. And you can see that for the highest numerical aperture of about 1.4, that's an oil immersion objective, you get about a quarter of a micron in the XY resolution and about 600 nanometers in the axial direction. If you reduce your numerical aperture to about 1, the XY resolution is not impacted that much, right? It's about 366 nanometers now. The axial resolution is only half as good as it was. It's now 1.2 microns. And if you go to a really low NA objective, like one that you would find on a 5X objective or so, then your XY resolution is actually still fairly decent. It's 2.5 microns, so about 10 times what you initially had. But it's now 53 micrometers in depth. So So all the cells that you have growing on the cover slip, for example, or that you plated there, are easily within within that depth range of that axial resolution, and you cannot discern any cells on top or underneath each other. Okay, so that's talking a little bit about here the 3D resolution, and I want to kind of summarize this with this drawing here, where we and you'll see this now in the next few slides. Reoccurring with different kind of resolution influencing factors here. So I want to introduce it here briefly. So we have this is a simulation now, what I'll show you. So this is our theoretical object that we want to image. So here's let's say a nucleus, which is uniformly labeled at a low intensity, and then a plasma membrane surrounding it here. And this is a cross-section through the midsection of that cell. And here we have our Poinsett function, this area disk. And now, as I described earlier, for each point of our structure, we'll use the Poinsett function as a stamp to predict how our microscope image will look like. So each point in the sample, if you zoom in here, will be blurred by this Poinsett function, and you end up with a blurry plasma membrane here in this case. And you can see that I chose the distance between the nucleus and the plasma membrane in such a way that you can barely resolve here this gap between the nucleus and the plasma membrane. So this is the diffraction limited resolution of our microscope at work now here. However, and this is now introducing into the second limitation of resolution. In practice, we don't have an image with infinitely small pixels. We have a limited number of pixels in our sample, and that's in our image. And that's based on the camera we are using most typically, right? So the camera can have 1024 by 1024 pixels. And that's actually how I simulated that image, right? And then here showing this at the original structure, not the image, you can see a fairly smooth and fine pixelation here of that image. But if the pixels would be 8 times bigger, right, then you only have 128 by 128, and this thing becomes quite pixelated here. And if the pixels are another 4 times bigger, then obviously the pixels become so large that you can't even distinguish here the plasma membrane from the nucleus. However, what I'm showing you here is not the pixelation of the image. This is the pixelation of the object. And that, of course, doesn't happen in practice. So what's more relevant is to look at the pixelation of our diffraction limited image, where I applied that Poinsett function. And that is done here. And you can see that you're actually not losing much information going from the 1024 image to the 128 by 128 image. You can still see this gap here that you could also see here. But you lose it when you go to this 32 by 32 pixel image. So at some point, the images get, the pixels get too large to really discern the features you're interested in. So what is the correct pixel size that you should choose now for your microscope image? And actually, before I go here, let me mention one thing here. And you might say, well, yeah, you're right. I can still see this gap here, but I still like the left image better than the middle one, because the left image looks nice and smooth and you see all the details, while here it's aesthetically not as pleasing. Yeah, that you might have a point there, but keep in mind that you will need tons and tons of more storage space on your computer to store the 1024 by 1024 images versus 128 by 128 images, right? This is 256 times less pixels than this one here. And what I want to also point out is that while this might look a little bit aesthetically less pretty, it actually contains the same amount of information. And I'll demonstrate this on these slides here. So I'm just showing the zoomed in area again of this finely pixelated image. And now I take this image and just add up 8 by 8 pixels into one new pixel. So that's how I generated this one. So I took this one and just resampled it at these coarser pixel sizes. And then I'm taking this image now and create 8 by 8 pixels out of each of these pixels again. I smoothen between these the neighboring coarse pixels to interpolate essentially that 1024 by 1024 pixel image. And as you can see, it virtually looks identical to the original one, right? So the details are fully recoverable from this 120 by 8 by 128 pixel image, because the image already was limited by the Poinsett function in its resolution before, and the pixels didn't make it worse. And just to show you the negative control here, if I do this with the 32 by 32 pixel image that I also showed on the previous slide, that is not recoverable, right? So here, this gap here between the plasma membrane and the nucleus stays obscured after I try to convert it back to 1024 by 1024. So this, the limit at up to which level you can still recover the details is described by the Nyquist theorem. And it essentially says that you can only recover details that are bigger than about 2 times the pixel size. So if you have a 150 nanometer pixel size, the smallest feature that you can reliably depict in that image is 300 nanometers of size. In practice, you better go not just a 2-fold factor, but let's say a factor of 2.5. And I think where this becomes completely obvious is if you think of a sinusoidal pattern, a sine wave, right? We have a peak every 500 nanometers. If your pixels are 250 nanometers or smaller, then you can have a pixel for the peak, then a pixel for the valley, and another pixel for the peak. They are 250 nanometers apart. 2 pixels will cover that 500 nanometer period. And so, but if your pixels become bigger than that, then you cannot resolve this, this periodicity of 500 nanometers of the sinusoidal pattern. So this is where this Nyquist theorem comes from. So we now have 2 resolution limits as microscopy users, the diffraction limit and the Nyquist theorem. And you need to keep both in mind if you want to know what you can resolve from your microscope images. So you saw the left half of this table already before, or the center part here, where I calculated the diffraction limited resolution depending on the numerical aperture. And now we have the Nyquist limited resolution, which depends on the pixel size and the magnification of your objective. So a typical modern camera has a 6.5 micrometer large pixel. If you have a 100x magnification, that means that each 6.5 micrometer large camera pixel corresponds to 65 nanometers in the sample. It's 100 times smaller. That's this 100-fold conversion here between the sample and the camera. So 65 microns now corresponds to 65 nanometers in the sample as pixel size. And now Nyquist told us that our resolution is about 2.5 times bigger than this 65 nanometers. And that's where I end up at 163 nanometers Nyquist limited resolution. So, but only the verse of the 2 resolutions is the one which matters, right? So in this case, with 100x 1.4 NA objective, you're not limited by Nyquist, but you're limited by diffraction now. You cannot resolve any feature smaller than about 260 nanometers in size. So if you now go down in magnification, and these are realistic objectives that you can find on your microscope. Let's say we go to a 25x 1.0 NA objective. This is one of these super fancy objectives used for intravital imaging or in vivo imaging nowadays, right? So large field of view, great resolution. Here in this case, you're actually limited by Nyquist because the image gets not magnified enough and the pixel size is now really the limiting factor. And it's not the diffraction limited resolution which limits you. So, and so on. You can see here that depending on the combination of magnification and numerical aperture, you end up with different limiting resolution values. The good thing is there are ways to overcome the Nyquist limit of resolution. You can just add an additional magnification if this is really your problem by getting a special tube lens which magnifies a little bit more, for example. And you can solve this to some extent if this is really your problem and you want to solve it. You cannot easily solve here the diffraction limited resolution. So that's something you always need to keep in mind. And to just bring this home one last time here, how the numerical aperture and the magnification now influence your image properties, right? So you have both of these written on your objective lens. So this is a 60x magnification, 1.40 numerical aperture objective. It's the magnification which describes how much an object is enlarged. But the size of the point function and the diffraction limit resolution is limited by the numerical aperture, not the magnification. And then just because of the Nyquist criterion, you need to take care of the magnification here. We already discussed this on the previous slide that you want to pick your pixel size to be 2.2 to 3 times below the points function size if you really want to take advantage of the maximum resolution that your diffraction limit performance of the microscope offers you. So if you now need to pick your objective for a particular application and resolution matters to you, pick a high numerical aperture for resolution and then choose the magnification based on the required field of view that you need to image the whole sample to be at the Nyquist limit. So next to Nyquist and diffraction, we have a couple of more softer factors which influence the quality of your microscope image. And I want to briefly here mention contrast and noise. Contrast is how much your specimen is essentially brighter than your background, normalized by the background intensity. So I think that's a fairly intuitive concept and something which I just want to emphasize here that background obviously is a problem here, and you want to reduce your background as much as possible. And then we have the topic of noise, and I'll have the next few slides actually talking about noise. So I don't go into detail here. And then the last thing is optical imperfections. Everything I've said so far assumes that you have a perfect microscope, but in practice, your microscope is not perfect, and that will also reduce the quality of your images. And we'll talk about that after the noise topic here. So noise. So you've seen this simulation before. This is now taking this structure, and this is the best image possible where we consider the diffraction limit and the Nyquist limit, right? So I showed you earlier that 100 by 128 by 128 for this particular image, that is sufficient to contain all the diffraction limited information. The problem, however, is that this here assumes that you have perfect signal and no background at all. In practice, though, you have only a limited number of photons, light particles, that are detected by your camera. And that means there's a stochastic noise component called shot noise in your image. We'll talk more about that in the next slide. And as you can see, you now get this graininess on top of your images from that stochastic detection. And if you have even less light to detect, this graininess becomes even more prominent. And it actually ends up to be a resolution limiting factor, right? Here you can still see this gap. Well, here it becomes kind of hard. And then if you go to this image, I would say it's impossible to see the gap between that plasma membrane and the nucleus. And it becomes even harder if on top of that now we have background, which will also have shot noise. And you can see how these different noise influences will really degrade your image quality. So where does this noise come from, and what can you as a user do to minimize it or avoid it? So in the next 3 slides, I think, go over here the different noise sources and what you can do about them. And actually, I forgot to mention it earlier, but text in green usually are recommendations for you from a practical perspective as a user. You'll see that in a second. So the shot noise is the first noise source. Shot noise is a very fundamental noise, which essentially has its source in quantum mechanics. And it really originates in the quantal nature of light. Now, the photons that we detect. I bragged about how bright a laser focus is, right? You have 10 to the power of 19 photons there. But now we are talking mostly about fluorescence images. And here we only talk about 1,000 photons that we get in each pixel. And at 1,000 photons per pixel, actually the stochastic nature of whether we detected the photon or not will actually play in here. It turns out that this stochastic nature creates here kind of the shot noise with a standard deviation, which is equal to the square root of the number of average photons that we would detect in that pixel. So if you have 100 photons in a particular pixel on average, then the standard deviation of the fluctuation of how many photons we will actually see will be square root of 100, so plus minus 10. Okay, so we'll get 100 counts plus minus 10 as shot noise. So the solution to that is, if you are shot noise limited, if you want to improve that, is to collect more photons. So collect as many photons per pixel as possible. And this becomes obvious if you look at the signal to noise ratio, which is the average number of counts divided by the standard deviation of the shot noise. And since if you put the square root of n term in for sigma here, then we have n divided by square root of n, which is square root of n. So the signal to noise ratio will become better and better the higher the number of photons is, right? It will scale with the square root of the number of photons. So how do you collect as many photons as possible? Well, there are many different strategies. The easiest and most obvious one is you increase the integration time. The longer you collect light, the more photons you will collect on a pixel. So you can change the integration or the acquisition time on your camera, for example. That's something you as a user can do with a mouse click, essentially. The second approach is that you can maximize your photon collection efficiency. Try to get as many photons reaching your camera as possible. So for that, you want to pick an objective with a high numerical aperture. Why that? Because the numerical aperture is the product of the refractive index with the sine of this collection angle of the objective. So the larger the NA, the larger your collection angle of that objective is. And the more light will actually be collected by that objective. You also want to have a look at the filters that you use to filter out your laser light and select your fluorescence light. And you might be able to pick filters which are a little bit have a broader spectrum to collect more of your fluorescence. And the last thing is you can pick a detector which has a high quantum efficiency. The quantum efficiency is your likelihood of detecting a photon that reaches the detector. And there, the general rule is the newer the detector, the better. So if you buy a new camera now, an SCMOS camera, it will have 95% quantum efficiency. If you compare that to a CCD camera, which is 20 years old, which you might have on your microscope, that only has a third or so of that quantum efficiency. The same applies to the newest generation of photomultipliers on your confocal microscope. They are much better than old ones. The third strategy is one which is also in your power, and that is choose a brighter labeling, right? Try to optimize your staining, obviously, to get as much light emitted from the sample as possible. And then the last two things are that you can increase the illumination intensity, turn up your laser power on the confocal, or turn up your lamp or LED light source on the wide field microscope. But be aware that that, of course, can introduce bleaching and phototoxicity. And then the last resort is that you can use larger pixels. So we were talking about Nyquist, right, that you need to watch out that the pixels don't get too big. But if resolution is not your concern, then it might make sense to actually not split your photons into too many small pixels, but actually choose pixels that are so large that you actually get a substantial amount of photons collected by each pixel. So that's about shock noise. And then the next topic here on the noise side is the detector noise. Yeah, I'll go through very quickly because it's not something you really do much about. The first one is dark counts. Some detectors accumulate significant background counts when you integrate for very long times. I'm talking about for a single frame, you record for seconds or so. You do this in some specialized imaging techniques like bioluminescence, but most of you will probably not experience this. It's a problem of older detectors also. So newer detectors are really not much suffering from these dark counts. If you have that problem, however, you can consider getting a better detector or cool your detector. It's actually has a thermal nature, this dark count. So if you cool your detector with liquid nitrogen or so, you can get down the dark counts. The second detector noise source is readout noise. This is more relevant in modern microscopes. And that happens when you convert the photons that reach your detector into the digital counts that you see on your computer screen. That digitization process is imperfect and adds some additional noise. Fortunately, the newest generation of detectors is far less vulnerable to readout noise. These SCMOS cameras are 10 times better or more than old CCD cameras. So my first recommendation would be, if that's a problem you face, get a new camera, buy an SCMOS camera. And the other option is that some of these cameras offer different readout modes, and there might be a readout mode for your camera which reduces the readout noise. In practice, fortunately, detector noise and readout noise do not play a big role on modern microscopes. So then the last noise source comes from the background. And here it can come really from all kinds of anything you can think of, right? It can come from the environment. In that case, turn off your room light. Shield your detector, your detection beam path, more particularly, from any external light sources. Your LED light source there might some light leak out at the side and reach your camera. Try to kind of shield that so that that doesn't happen anymore. It might come from the microscope, where I guess that what I just was saying. A laser or lamplight can directly reach the detector. So pick carefully your optical filters that filter out the lamp or laser light from your fluorescence. And sometimes the immersion media or some optical elements you might have put into the beam, they might actually create some autofluorescence, and you might want to think about replacing those. And then the last background source, and that's in practice probably the biggest one, is your sample itself, right? It can be autofluorescent. A common strategy there is to go to longer wavelength. Autofluorescence is often in the green spectrum, so going to red often helps. And also you can optimize your filters to block as much of the autofluorescence as possible. And then of course, background can also come from unspecific staining, where your antibody, for example, was used at a too high concentration, and now it sticks not only to your protein of interest, but everywhere. And there the solution is to optimize the labeling protocol. So I'll want to spend the last, I'm going a little bit over. I'll try to finish up by 12, Diane, is that okay? Okay. So the last topic here when it comes to image quality and resolution is related to these optical imperfections. And for that, I need to explain briefly the refractive index, because that is at the source of this problem. So the refractive index is a property of the glass or the medium you go through with your light. It essentially describes how much the speed of light is reduced when you go through that glass. So the speed of light I mentioned earlier, this 3 × 10⁸ meters per second, is the speed of light in vacuum. However, in glass, it's about 30% reduced. And because we need to divide that speed of light in vacuum divide by, let's say, 1.5, which is a refractive index for glass, roughly, and then you get that new reduced speed of light in the glass. What the result of that is, and that's the problem in microscopy now, is that it will change the direction the light travels. So if we have an emitter of light sending out these waves here, and they hit, let's say this is an air, and it hits an air glass interface, and then it goes into the glass, because the speed the waves are traveling is reduced, the direction the waves are traveling in is changed. So if you just follow here the kind of the waves, and then you see where they go next, right? You see that they have this this bend now. And this is, you can calculate this quite nicely here with Snell's law. You might have seen that in a physics course you took recently or years ago. This, of course, is a very useful property if you build a microscope, because this is how we build lenses and actually direct light towards our sample, and we collect the light and all that. But it can also be a problem when it comes to our sample, because not only glass has a different refractive index, but also water or PBS or whatever your sample is embedded in. They all will have different refractive indices and will experience this effect that they refract the beam of light, they change the direction of light going through. So let me show you one practical problem where there's really, where you can see that in daily, essentially, when you work with a microscope. And that is if you focus with a confocal microscope into a sample. So here I'm assuming the objective sits on the left, and I only draw here the directions these rays of beam the light is traveling through. The red line in the previous plot here, right? This red line is now here shown in these black lines. So you have light focusing here into this common spot, which is the focus of your confocal microscope, for example. And I'm assuming here that we go through some immersion medium or a cover slip, which has a refractive index of 1.52, and the sample that's embedded in the same refractive index. And then these rays of light will go through unobstructed, just straight through, and you get this perfect focus. That's what your oil immersion objective is built for. That's what the oil immersion objective assumes you actually use as a sample. However, if you now replace the refractive index of your sample with water, and or PBS or so, and you now have an average refractive index of 1.37, then these rays of light, when they hit the glass PBS interface here, will change their direction slightly depending on the angle they are hitting that interface at, right? The central one here, which hits the surface in a normal orientation, will not change its direction, but the others will. And you can see that they don't reach the same focal point anymore. You degrade your focus quality. And this becomes even stronger when you move this interface further away from the focus. So now we focus deeper into the sample. Let's say this was 30 microns deep, now it's 100 microns deep or so. And this effect gets even worse, right? So if you focus with an objective which is built for particular immersion medium into a medium of a different refractive index, you'll get this refractive index mismatch effect and image degradation. Here you can, for example, see an image how this looks like. You have here the well-corrected image, and here you have the image when you have that refractive index mismatch not corrected. In this particular example that is shown here, this was actually not an oil objective focusing into a aqueous sample, but it was a water immersion objective, which has a correction collar for the cover slip thickness. These objectives are already built to correct for this refractive index mismatch, assuming a cover slip of a certain thickness. And this cover slip thickness is written down here on this correction collar, and it goes from 0.11 millimeters to 0.23 millimeters. The standard thickness that number 1.5 cover slips have is 0.17, and that's the cover slips you should buy. And here you can see what happens if you have not adjusted the correction collar. Then you see kind of a much blurrier image than when you have corrected the correction collar of this objective. So in summary here, I'll show you this overview of what factors you should consider when choosing an objective. We talked about the magnification and the numerical aperture. We talked about the refractive index mismatch, which means you should really pick an objective with the right immersion type so that the refractive index is matched. And you should correct the cover slip correction collar if your objective offers that. Other factors are if you do polarization, DSC, or phase contrast imaging, you need to choose an objective which is suitable for that. You might also need to look into different corrections. Some objectives are built for very large field of view, flat field corrections. Others are built to work with many different wavelengths of light at the same time. And then there's the working distance. Some objectives only can penetrate 100 micron deep into a sample before they bump into the sample, and you can't go any further. Other objectives can go 2 millimeters or 8 millimeters into a sample. So if you are working in intravital microscopy in particular, this becomes a big concern. Okay, and you can find this information, essentially all these specifications I'm listing here, quite well on the manufacturer websites that build these objectives. So that's a great resource here. So then the last few minutes I want to spend on here chromatic effects, so color dependent effects, and they are also related to the refractive index. And the problem with the refractive index is that it's dependent on the wavelength. So a different wavelength, the light will actually experience a different refractive index and therefore also different amount of refraction. And we know this from the prism, right? The blue band is refracted more, and the blue light is refracted more than the red light here. And we also know it from the rainbow in daily life. But we also unfortunately have this problem with lenses. A lens will also refract the light and will refract the blue light more strongly than the red light. And that means that the focus of our confocal microscope, for example, but the same applies to the wide field microscope, just the other way around, that the focus appears at different depths in the sample. The objectives are usually corrected to a large extent to correct for that, but they will not be perfect. And here, for example, I'm imaging a 3-color stained fluorescent particle. So all the 3 colors should be on top of each other. But as you can see, the blue spot is actually deeper in the sample than the green and the red spots, even though they originate from the same particle. So the solution, so first of all, I want to make you aware that this problem exists because it can lead to colocalization artifacts. If you're not aware of it, you might think that the blue staining is actually at a different location than the green and the red one. Here I know it's not because the particle was triple stained, but you might interpret this as biological results, right? So if this is an analysis you want to do, the solution is here that you actually calibrate for it. You measure, you take these fluorescent beads with 3 colors and you image them with your microscope, you measure the distance and you correct for that distance the shift in your image processing later. Okay, and then I have 2 slides left here. And now we are really coming into very practical advice, more on the image analysis side, and then talking about multicolor imaging. And this is a classical mistake I've seen users on microscopes do fairly regularly, and can also lead to artifacts. And that's why I would like to pointed out here. And that's the problem of saturation. So I would argue the image in the top here is a much better image than the image in the bottom. And the reason is that the bottom image is separated. I've lost the details here in the brighter objects. For example, here the mitochondria in green, I can still see them as hollow objects because it was a membrane staining used here, an outer membrane staining, while here they are completely merged into uniform green dots here. And then I've also lost the very weak red microtubules here. These ones here, I can see quite well here, while here they are filtered out from my image. So I've misrepresented the original distribution of my fluorescent probes here in my saturated image, right? I lost the weak signal and I lost the contrast at the very high intensities. So where does this come from? So the problem is that we have a certain spread of signal in our coming from our sample, right? There will be very dim areas and there will be very bright areas of our signal. This will be very bright emitters. But our detector has a limited range, and we as users decide what range we detect, right? So and we have lost potentially the counts at the bottom and counts at the top, we call overflow and underflow, by choosing the wrong detection range here. So you can adjust this detection range by choosing the right amplification of your detector, or by choosing the right laser power or lamp intensity, so that you are not creating too much overflow. And you can also adjust the offset on your detector to make sure you're not creating this underflow problem. The problem is that you are not safe after you took your image. In your following image processing, you can also create these saturation effects by adjusting the contrast and the brightness in your image settings wrongly. So even if your original camera image or photomultiplier image with a confocal microscope looked good, if you then analyze it in the wrong manner or display it in the wrong manner, you can create these saturation effects here. So image processing, it easily can happen, but even in your final PowerPoint presentation, it can happen accidentally if the projector of the seminar you're using is wrongly adjusted. The most important step, however, is to get the detection process right, because if you can redo the image processing, and you can certainly redo your PowerPoint presentation, but you might not be able to redo your imaging session of years of work if you later find out that you accidentally saturated your data there. And then the last topic I want to talk about is that of crosstalk and bleed through. That's a multicolor imaging issue. And it arises here from the problem that you have different fluorescent dyes which overlap in their spectrum. So here in the left column, you see a good image where the green and the red channels are nicely separated from each other. While here in the right column, you see that the green channel was bleeding into the red one. So the mitochondria here in the green channel are also showing up overlaid on top of the red microtubules here in the red channel. And the problem here is that this can lead to interpretation artifacts, right? So you might think that whatever red label you used is not only associated with microtubules, but also associated with mitochondria, because the mitochondria appear in yellow here, right? And that in this case would be wrong. So it's, again, you end up with co-localization artifacts. So the solutions here is, and we cannot go much in details here right now, but I'm happy to discuss this in more depth in the following hour, is to use dyes which are spectrally well separated to optimize your filter settings and the laser powers and detector gains, so that not one of the dyes is amplified much more than another one. And a very good approach, if you can, if the microscope offers it, is sequential scanning, where you first take the green image and then the red image, and you do it in a interleaved manner. That way, the green laser will not excite the red dye, and vice versa. And if you couldn't fix it on the imaging side, you can still use a spectral unmixing algorithm later on to separate these overlapping spectra if you know how much bleed through and crosstalk you had between the different channels. I know this was a tour de force, but I thank you for staying with me, and I end here. And I hope this was useful to you, and that if it wasn't clear here from this first round of explanations, maybe it helps to look at it later. But I'm more than happy to answer any questions you might have in the next hour or so. And I end here. Thank you.
Thank you very much. You know what I just learned is I think I wish you were my physics teacher 40 years ago. And also that I knew some of these details as I was doing imaging across a process. You know, you go to a core facility and you use the core, but you might not know any of those details to sort of help make educated choices as you're doing your imaging. So that was really terrific. Thank you so much. We're going to open it up to anybody who has questions, and you can just unmute yourself and ask a question if you have a specific question that you want to ask. You're watching. Many people are jumping off now, so we might not have too many questions, but please.
That's perfectly fine. If there's only one person, right, who has any questions.
Yep.
Joerg, this is Ken Dunn. First of all, can you hear me?
Hi, yeah, hi, Ken.
Good, great. Anyway, I was wondering if you could comment on how the trade-off between Nyquist resolution and speed and signal and noise changes when you start looking at living cells. So in other words, everything gets harder as you start putting more light in and you try to integrate longer and all that other stuff. So can you talk about that a little bit?
Yeah, you're absolutely right. This is a good point. Yeah. So when you image live samples, you have often two problems. The one is speed. You need to be able to image fast enough. And second, which is probably the bigger issue, is phototoxicity. You cannot afford to illuminate the sample with too much light before the sample dies, right? So now think of this phototoxicity issue as a dose question, right? So let's say you can afford the sample to emit 1 million photons before it shows signs of phototoxicity. So you now have 1 million photons that you have to spread over your pixels. So if you have many pixels, that means each pixel will only get very few photons. And that means each pixel will have a very high shot noise. And that means each image will look very grainy, and maybe so grainy that you can't analyze it at all. So in this case, you might need to sacrifice resolution because you say, I just plainly cannot afford to image here at the Nyquist limit, or certainly not better than the Nyquist limit, what people like to do because you get these smoothly looking images. And you need to essentially choose a pixel size, which is maybe 1 micrometer or so in size. But that way you get enough counts in each pixel to analyze it quantitatively later without creating too much phototoxicity. And of course, I was explaining this for a single image, but now think of this imaging 1,000 images, right? So a long imaging series, this really becomes a quite dominating factor. But I mean, Ken, you have done a lot of live imaging, so maybe you should give your perspective.
Actually, I have another question for you. This is a more detailed question, I guess. A more difficult question is, how do you evaluate phototoxicity? I mean, it's something that you will frequently read a paper where someone's claimed that the imaging had no influence on the biology they're looking at. And that may or may not be true. In a way, it's very easy. You know, looking at the microscope from across the room, you didn't see any evidence of phototoxicity. You know, it's a question of what you look at. I was involved in an experiment once where, as a core facility, people come in and they do studies, and he was interested in whether or not a specific pharmaceutical reagent induced reactive oxygen release. And so we were there, and we're imaging this one field over time, repeatedly imaging it. He adds whatever it is to the stage, and we keep on imaging, and everybody's really delighted because the biosensor reading reactive oxygen got brighter and brighter and brighter and brighter. And there was great celebration in the room because everybody decided that the drug had worked. But when you zoomed out in the confocal image, what you discovered was that it was literally just the one region that you'd illuminated that had responded. And no one was interested in that in the room at that time. But the point is that a lot of times you have to evaluate what are you saying? I mean, sometimes cells will actually bleb and blow up. That's a pretty crude indication of phototoxicity. So how do you, in your experience, what do you think is a good way of determining whether or not the imaging is influencing the event?
I think